Welcome, everybody, to the next episode of the Cannabis Review. I'm delighted to be joined in this episode by Nicholas Milne, who's the co-founder and CSO at Danish-based company Octarine. How are you keeping today, Nicholas?
Good, thank you. And yourself?
I'm very well. Thank you very much for taking your time to do this.
Sure.
Do you maybe want to give everybody an overview of your position in your sector and how you guys came about to found Octarine?
Yeah, sure. So my background is, and my co-founder's background is in yeast fermentation and synthetic biology, which basically means we engineer the genome of yeast to produce new and interesting compounds by fermentation. And basically it's akin, I don't know if you know, but the yeast is this microorganism that makes beer and wine and bread and all these wonderful things we know and love. But by playing around with its genome, you can get it to produce other interesting compounds, basically in the same apparatus. So our fermentation process looks very, very similar to a process you'd see at a brewery or at a drug manufacturing facility. What we do differently is we get that yeast to produce compounds that it wouldn't natively produce. So we work within the psychedelics value chain to manufacture psychedelics. We see a lot of difficulties in extracting psychedelics from their native host, particularly psilocybin from mushrooms. Mushrooms aren't the easiest organism to cultivate, and particularly they're very difficult to standardize. So we see fermentation as this really nice middle ground where we can get all of the sustainability benefits of fermentation, but we can offer a much more consistent and stable supply of these compounds. And importantly, cost. One of the big advantages of yeast fermentation is the cost that we can produce these molecules. And just as a rough input, we're aiming to be able to develop or to provide GMP grade psilocybin for dollars per gram, and perhaps even lower than that. So the cost benefits are really, really significant.
Okay, can we maybe break down the difference between using fermentation and biosynthesis for cannabinoids and for psilocybin? Obviously, psilocybin, we'll start with first. The quantity of psilocybin per mushroom or per plant is not replicable over and over again. So large-scale cultivation is not really a solution for large-scale drug discovery and development. Is that correct?
Well, it is possible. We just argue that it's not very efficient. So as just a rough example, psilocybin producing mushrooms, they produce psilocybin in the range of about 1% dry mass. The problem, though, is that on top of producing psilocybin, these mushrooms also produce a range of structurally related compounds. So these are the intermediates, byproducts, degradation products. And from a purification point of view, you have to invest a lot of additional resources to separate your compound, which in this case is psilocybin, from the myriad of other structurally related compounds. So it does add a further complication step. It's also, and you know this if you go to the supermarket and buy mushrooms. Mushrooms are hardy creatures. They have this tough outer layer. They're very difficult to kind of burst open. And so you have to invest a lot of resources in order to get the psilocybin out. In contrast with our fermentation method, our yeast, it grows sensationally well in large-scale fermentation. It simply secretes the psilocybin into the surrounding media. So from a purification point of view, that's much easier. And I think most importantly, our strains, they only produce psilocybin. They produce nothing else that even looks remotely similar to psilocybin. So from a purification point of view, that also makes it considerably easier.
And so we can pretty much say the future of this ingredient side of both psilocybin and rare cannabinoids is going to be fermentation for the environmental impact that it has, the scalability it has, and as you said, the dollars per gram price. Do you see this over the next 10 years being the dominant player when it comes to this side of the ingredient market?
Yeah, this is how we see it. And yeast fermentation is used to produce already a surprising number of the ingredients in your daily lives. It's used to make vaccines, pharmaceutical drugs. It's used to make food ingredients, biofuels, bioplastics. It's already out there in surprisingly large quantities. I think the area where it hasn't quite penetrated to the extent as other industries is in small molecule pharmaceuticals. So we see this as an emerging application. And when we looked at psychedelics, we see, the industry is being built mainly from the ground up, right? This is not an existing industry. It's being built from scratch. And we see this as a really good way to incorporate sustainability from the outset. instead of having to develop an unsustainable industry and later go in and improve it. We see an opportunity to use that from the get-go, and we see fermentation as the most realistic way of achieving this.
And now let me move on to the cannabinoid side of the industry. Obviously, everybody knows about CBD and THC as the most prominent cannabinoids, but we all know that there's over 120 plus cannabinoids now. When it comes to those rare cannabinoids that are available only in a minute percentage, is the same process applied to psilocybin, or is there a different technique that's used to extract these rare molecules that are at small scales?
So in theory, the story is very similar, right? The cannabis plant produces 120 different compounds, most of them in incredibly minute amounts. How do you gain access to those? But I think a key difference is that cannabinoids are notoriously complicated molecules, and the way that they're biosynthesized is extremely complex and far more complex than most people realize. And so actually, as a production system, the cannabis plant is, in our opinion, probably the most efficient way to get your hands on these molecules. And what we've seen in the last few years is that we've seen, in particularly breeding techniques, allowing us to gain access to some of these more rare cannabinoids. So in contrast to psychedelics, we actually see plant-derived cannabinoids, and particularly of rare plant-derived cannabinoids, as actually being the major driver of the cannabinoid market. Where we fit into this value chain is, so we don't have an interest in manufacturing cannabinoids. We think the plant is doing a fantastic job. It's cheap, it's readily available. The problem we see with cannabinoids is their properties, and anybody who's worked with cannabinoids knows that issues around their solubility, their stability, and particularly their bioavailability are really rate-limiting steps. And so actually what we're doing in this industry is we're using fermentation and, again, sustainable bio-based approaches to convert natural cannabinoids into new and improved compounds with these improved properties. So with this very simple fermentation-based method, we produce cannabinoids that are fully water-soluble, are extremely stable, and have very high bioavailability. And we see this combination as really the way forward, where the cannabis plant produces the molecules, we improve them, and that's the product that makes it to market.
We hear a lot of talk about the full spectrum effect of all the cannabinoids that come from a plant together. Is the future of this industry, if we're able to biosynthesize molecules individually, creating new formulations that may not necessarily already exist in the plant, but are more advantageous for whether it's inflammation or whatever ailment it may be?
Yeah, so we do see this talked about a lot, the idea that multiple cannabinoids and perhaps even cannabinoids mixed with terpenes synergistically work together to have an improved effect. And that definitely seems to be something that's happening. I think when we talk about things like full spectrum versus individual compounds, I think that's more of a reflection that we don't fully understand how all of these different molecules work together. And so until we really understand that, the strategy is just to mix everything together and assume that they'll all work together. The way that we see it is that in the future, I think we'll understand in much more detail how specific cannabinoids interact together. And we do see formulations where perhaps it's a mix of two cannabinoids and maybe a terpene that gives the overwhelmingly the therapeutic effect. So instead of having to mix, 100 different cannabinoids together, you can just mix 2 or 3, perhaps with a terpene, and that is your therapeutic product.
Do you guys at Octarine work on your own formulations, or are you singly just focused on distribution? We create the molecules and we bring them to companies who then work on formulation. Is there a big need and a big want to get into the formulation creating side of things?
So we're firmly a business-to-business company. So we work with consumer brands, but also pharmaceutical companies or drug development companies to advance products made by fermentation down the value chain. So in the case of something like psychedelics, we would work with, mainly with pharmaceutical companies to take lead candidates into the clinic. On the cannabinoid side, we work with consumer brands who are really looking at specific cannabinoids to get into consumer products. So for example, consumer-facing company A might be really interested in a water-soluble version of CBD. Consumer company B might be really interested in CBG, and we develop the manufacturing process to get those products into market.
How important is the teeter amount when it comes to the competition between other companies in your market? Is it a case of whoever gets the highest grams per liter per hour is the winner of the race, or is there a scale of purity that can be more advantageous than going for the high quantities?
Yeah, so at the end of the day, cost of goods is the most important parameter, right? How much does it cost to produce a kilogram? Titer is a really important aspect of that, but it's by far not the only important thing to consider. So we consider, yeah, so titer is a really big thing, but actually the most important factor when it comes to cost is the cost to purify. How much do I have to invest to separate my molecule of interest from the sometimes complex fermentation broth? And that, I think, is where, at least in our point of view, is where most of the R&D investment is put in how to purify.
And is choosing the correct substrate a difficult task, or is it the same for most companies?
So, I mean, so sugar is by far the most commonly used bulk ingredient. It's incredibly cheap, particularly compared to the cost of the product, right? If we're producing psilocybin at dollars per gram, glucose is cents per gram, and maybe even a little bit less than that. So I would say that, at least on the psychedelic side, not many companies are thinking more complicated than just adding sugar. In the case of cannabinoids, I actually think it's a little more complicated. It's, as I said, producing cannabinoids is notoriously difficult, and producing it from glucose or other sugars is actually complicated as well. So I know that many other companies are using sort of intermediates of the biosynthetic pathway, so things like hexanoic acid, which is more expensive than glucose, but it does make the production process more efficient.
When it comes to the biosynthesis of cannabinoids, is everybody starting with olivethyllic acid and geranyl phosphate and combining those for CBGA, or is there an easy entry point to start with THC or CBD?
So CBD and THC are, for the most part, the end of the biosynthetic pathway. So there's not that many things you can convert these cannabinoids into. There's a few degradation products and things like that. The first sort of common intermediate is either hexanoic acid, or in the case of producing the kind of big three, THC, CBD, and CBD, CBG, THC, then olivethyllic acid is kind of the starting point. Wherever you start, there is a cost factor to consider, right? Glucose is cheapest, hexanoic acid is more expensive, olivethyllic acid is more expensive again. So it really determines where your cost of goods comes from. I think the goal of synthetic biology is always to produce complex molecules from very, very simple starting substrates. So any synthetic biologist always prefers to start from glucose or something similar like that as the starting point, because that's always going to be the cheapest process. But if there are difficulties with the biosynthetic pathway, like in the case of cannabinoids, then an alternative could be something like one of the intermediates.
Would we be right in saying that the future, especially in Europe, of the ingredient side of whether it's food and beverage and cannabis or beauty and makeup or drug discovery development, is going to be led by the biotechnology companies, that the scalability and ability to just extract from plant matter won't be able to compete?
I think it really has to be taken on a case-by-case basis. You know, again, as a synthetic biologist, we wished we lived in a world where we could outcompete every other process, but there are many examples where plants are just cheaper. And so I don't think you can just apply a one-size-fits-all approach. You really have to look at the individual compound. You have to look at the market that it's being applied to, and really holistically look at the best way of bringing that product to market. In many cases, the plant wins, and that's okay. We believe that for cannabis, that the plant is going to win over any other technique, but there are many other cases where different molecules, it makes financial sense to go for something like yeast fermentation.
It's been absolutely fascinating talking to you, Nick. I wish we could have a longer episode, but we're nearly at our 15 minutes time now. For anybody who hasn't ever checked out Octarine, I highly recommend it. The website is below and it'll be in all the descriptions. But for now, Nick, thank you very much for your time. It's very much appreciated.
Yeah, thanks a lot. Nice to talk to you.
Until next episode, everybody.